Okay, I hope everyone is able to see the screen as well. Okay, now let's continue it. So now everyone listen what exactly we have to study in the biochemistry. Okay. So whether it's a botany people or the zoology people, why biochemistry is very difficult is in case of a biochemistry in your JL or the DL, we will get the questions, most of the questions will be direct based questions only, but some questions will be from the depth of the concept. So that is the reason in biochemistry everyone has to focus on the concept, okay, rather than remembering the things. So in biochemistry there are very less information to be remembered and more information to be understand. What exactly we have to study in biochemistry? See, since today is the first class, I will go little slow. From tomorrow onwards we have to go little faster. In case of biochemistry, we have to understand complete carbohydrate metabolism. We have to understand complete carbohydrate chemistry. Whereas we have to understand, see, when I use the word carbohydrate metabolism, we have to study many different metabolisms. It is not like only glycolysis, Krebs cycle, electron transport chain. We have to study different type of mechanisms. Along with the carbohydrate in our syllabus, it was very clearly mentioned lipid metabolism also, as well as the lipid chemistry also, and as well as enzymology also we have to study, right? See, for the botany people only this much is there, okay, but the zoology people and see this only this much syllabus is there for the botany JL, but the botany DL people and zoology JL people as well as zoology DL people has to study the proteins also, has to study the amino acid also compulsory for the exam, because amino acids are also extremely important, which is linked with the molecular biology, and the questions will be expected from the molecular biology as well. Okay, now let's continue. So please do not disturb in the middle. So I will explain it little slowly and effectively. Please try to understand it correctly. So carbohydrate metabolism. Okay. So friends, what is metabolism first of all? Whether it is a carbohydrate metabolism, protein metabolism, or amino acid metabolism, or lipid metabolism. First of all, what is metabolism? How can you define the word metabolism? See, metabolism is a chemical reaction which is taking place in every single cell in our body, correct na? So in a simple words friends, metabolism consists of catabolism, metabolism consists of anabolism, and every one of you know very well catabolism means what friends. Always keep the point in your mind. Catabolism means we are breaking down something. And what is anabolism? Anabolism means we are building up something. Catabolism means what? We are breaking down something. We are anabolism, we are building up something. Now listen, all of you try to understand the concept correctly. Suppose friends, we have taken a food. Whatever food contains, our food contain carbohydrates, our food contain proteins, our food contain fats. Every one of you try to understand me correctly. When we take a food, whatever the biomolecules that are present in the food, they are getting breakdown. Is it or not? When they get, now the question is very clear, sir, why we have to break down them? That is very important point. See, the breaking down of the biomolecules only we can call the oxidation. Oxidation words represent breakdown, or you can also call it as a catabolism. Now question is very clear, sir, why we have to break down all the biomolecules? We are breaking down all the biomolecules to produce energy. Remember my words, in the catabolism we will produce the energy, and in the anabolism we use the energy. Okay, whatever the food that we take, from that particular food we produce energy. Now the question is very clear, what we are going to do with this particular energy? Friends, every one of you remember my words. In our body, human body, there are 37.2 trillion cells are present. How many are there? 37.2 trillion cells. But the question is very clear. All the 37.2 trillion cells present in our body, if they want to work, compulsory there is a requirement of energy. Without energy, none of the cell in our body never ever work. If they want to work, they require the energy. So whatever the function that we performed, if we want to do the muscle movement, if we want to do any kind of a transport mechanism, if in our body any kind of a synthesis wants to occur, any function performed in a living cell, there will be a compulsory requirement of energy. So what you have to understand, when I eat the food, I will break down all the food, whatever I have taken, I will produce the energy. And what I am going to do, I am using that energy to perform all the functions in my body. So when I am performing some functions, what is happening? Everyone look into this. This ATP is getting break down into what? ADP and PI. So that's why ATP and PI are said to be what? Hydrolysis products. Why did I use the word hydrolysis, friends? Because I am breaking down the ATP into ADP and PI. Whenever I break down, what is happening? There will be a release of energy. That energy only we used to perform various functions. So metabolism include oxidation, which is called catabolism, building up something which is called anabolism. Together only we can call it metabolism. Whomever joined, please join just now, please continue. This is only the first slide. Now see, everyone remember what are the various stages of metabolism. See, biochemistry will be little difficult in the first few days. After that it will become very easy, so focus. Now, see friends, remember everyone, whether you take, suppose example, just imagine, today I had a biryani. Imagine today I have a biryani. What does this food contains? It contains polysaccharides, which is sugars, and it contains lipids as well as it contains proteins. Is it or not? But one point every student has to understand, friends, whether it is a polysaccharide, whether it is a lipid, or whether it is a protein, what is our ultimate goal, friends? Our ultimate goal of all these is we have to break down them and we have to produce energy. But the point is very clear. The way they produce the energy is little different, but only they are having a common goal, that every biomolecule is going to give rise to the energy only, but by using different pathways. Let me tell you what are those pathways. Look into this slide, very important slide it is. Polysaccharide. See, suppose we eat rice. What is rice, friends? Rice is the starch. Correct, na? Now you tell me, when you eat the rice, do you get the energy directly from the starch? No. First, what is going to happen? The starch, which is a polysaccharide, has to compulsory break down into oligosaccharide, disaccharide, ultimately monosaccharide. Why? Because monosaccharides only can easily travel in the bloodstream. They are the one which is simple forms. They are the one which can produce the energy. So what are the various polysaccharides? Glucose, fructose, galactose. Now you can ask me a question, sir, why can't we get the energy directly from the fructose and galactose? Because friends, glucose is the one which can travel in the bloodstream very easily. Most of the tissues in our body, they require the energy in the form of glucose. You take an example, brain. Do you think brain will use fructose? No. Do you think brain will use galactose? No. Brain always use the energy in the form of glucose only. Is it or not? So what should happen, friends? Whatever the carbohydrate it may be, finally it has to be converted into glucose. Now you all know very well, I will tell you in the upcoming slides what is going to happen now. The glucose ultimately converted into pyruvate. Now the question is, friends, how the glucose is getting converted into the pyruvate? Glucose is getting converted into the pyruvate by a process called as glycolysis. We will study it elaboratively. But then later, pyruvate is going to be converted into acetyl CoA. Then acetyl CoA converted into acetyl CoA entered into the citric acid cycle. Then it gives rise to abundant amount of energy. Now, friends, this is the fate of a carbohydrate. Now, in our food, not only carbohydrates, right? There will be a presence of a proteins. Now, friends, you can't use the direct proteins to produce the energy. That is not possible. So what is going to happen, friends? Proteins are getting converted into amino acids. Now every student understand. Here I told you that, friends, glucose undergoing the glycolysis process and giving rise to the pyruvate. Similarly, friends, amino acid undergo what kind of a process, friends? We can call the transamination. What do you mean by the word transamination? Transfer of amino group. Most of the students are getting confused between the deamination and transamination. Deamination means, friends, you're removing the amino group. I am not removing any amino group. In the amino acid, I am just transferring the amino group. Upon transferring that amino group, you see here, amino acid convert into pyruvate. Some amino acids are directly going to give rise to the acetyl-CoA. Once again, acetyl-CoA enter into the citric acid cycle and produce abundant amount of the energy. So proteins are also ultimately what? Giving rise to ATP only. But the process is different, which we can call transamination. Same, our food also consists of what? Our food also consists of lipids. Now, friends, of course, you know very well, lipids can't break down very easily. Lipids are very large molecule. So first, initially lipid break down into fatty acid and glycerol. Once again, glycerol converted into pyruvate. Pyruvate converted into acetyl-CoA. Of course, acetyl-CoA enter into the citric acid cycle. Now, friends, remember, breakdown of glucose, what I have called, I call it glycolysis. Breakdown of amino acids into acetyl-CoA, what did I call? Transamination. Similarly, friends, breakdown of fatty acids to give rise to the acetyl-CoA, that is what exactly we can call beta oxidation. I want you to remember the name. Steps are very important in biochemistry. What? Breakdown of fatty acids into the acetyl-CoA, we can call beta oxidation. Oxidation means what? Breaking down. Beta, why we are called beta? Because the breaking down occurs. Breaking down starts from the beta carbon. I will tell you later. That's the reason we can call beta oxidation. So by the beta oxidation also, undoubtedly it is going to give rise to the acetyl-CoA. Acetyl-CoA enter into the citric acid cycle, produce ATP, NADH and FADH2. Now my question to all of you is very simple question. My question is, what is the, listen, think and answer. Everyone has to type it. Right or wrong, doesn't matter. Everyone type it. What is the key intermediate molecule in the carbohydrate metabolism? Not carbohydrate, in any metabolism. What is the key intermediate molecule? You tell me in this particular metabolism, which is the key intermediate molecule? Undoubtedly acetyl-CoA. Is it or not? Because, friends, whether it is a carbohydrate, whether it is a protein, whether it is a lipid, under any conditions, everything is converting into what? Of course, everything is converting into acetyl-CoA, not the pyruvate. Because the pyruvate is obviously gonna convert it into what? Acetyl-CoA. Don't get confused with the point. Okay, absolutely true. So, friends, now listen, everyone of you, listen, everyone. Till the end of our biochemistry, remember everyone, till the end of the biochemistry, we keep on using ATP, NADH and FADH2 throughout the lesson till end of biochemistry. So that is the reason every student has to know what is ATP, what is NADH, what is FADH2, because entire lesson we are going to study this. So you should not get confused. You have to study biochemistry in an organized manner, then it will become very easy. Now ATP, adenosine triphosphate. So every student remember, ATP nothing but what do we can call the energy precursor, we can call it energy currency. So if we want to purchase something, we compulsory require money. So money will be the energy currency for us. Similarly, inside the body, if the cell wants to function, cell require currency. What currency? Energy currency. What was that energy currency? ATP. It is the energy form stored in the cell. How we are going to get the ATP? I already told to all of you friends. We are getting the ATP. How? by the oxidation of a food. What do you mean by that? By the breakdown of the food. Now, what does the ATP consist of? You only look into this picture. I am telling you whether it's a JL or DL, you people no need to remember any structures. I will tell you what structures to be remembered. You focus on only those structures. You no need to remember all the structures. Don't worry at all. Now, if you look into the structure of ATP, look into this. What does it contain? It contains sugar, which is a ribose sugar. It contains nitrogenous base adenine, and it contains three phosphate groups. How many phosphates are there? Three. So alpha phosphate, beta phosphate, gamma phosphate. Sometimes instead of calling alpha, beta, gamma, we can call monophosphate, we can call diphosphate, we can call triphosphate. Anything is same. So we generally call it as ATP. So now friends, the question is very, very clear. I am continuously saying that ATP is going to give rise to the energy. So how much energy? That I am requesting you all the time remember with respect to the kilojoules, because in the exam they will always ask in the kilojoules. So friends, and let me ask you a very simple question. I am saying that ATP is energy. You have to remember from where the energy is coming from. That's also good question. See friends, the energy is always in the form of phosphate group. Phosphate is the energy. I want you to remember that point correctly. When I break down the ATP into AMP, I will get the lot of energy. But the point is, friends, I can't break down the ATP directly into AMP. First, I will break down the ATP into ADP. ADP is what? Adenosine diphosphate. AMP is what? Monophosphate. There's a single phosphate. When I break down ATP into ADP, 31 kilojoules of energy will release. Similarly, friends, when I break down the ADP into AMP, another 31 kilojoules will be released. So friends, in a simple words, when I break down one ATP into AMP, there will be a release of 62 kilojoules of energy. Similarly, when I want to farm, I mean to say, I want to synthesize ATP from the AMP. Here also I can't synthesize directly, right? What I am going to do? First, I am joining AMP with the ADP. Sorry, I will form AMP with ADP from the AMP. Then I will convert ADP to the ATP. Then I have to use 62 kilojoules. In simple words, when I break down ATP to AMP, 60 kilojoules of energy is released. But when I am converting AMP to ATP, 62 kilojoules of energy is required. That's all. But the energy will be remain same. No need to get confused with that point. Okay. So now, so look into this. First, I am breaking ATP to ADP. Later, I am breaking ADP to the AMP. As I already told you, friends, what not? Every single function happening inside the cell, there is a requirement of energy. And listen, whenever you're solving a question, please don't get excited and don't be hurry. You know, calm mind or a peaceful mind always give the right answers. Okay, slowly analyze, don't compare with others, slowly answer the question. Okay, everyone type the answer. Suppose I want you to type answer like this. Suppose option 1 will be the C and D, option 2 will be the A and B. Type everything at a time. Take 1 minute, take 30 seconds of time, and everyone answer the question. Match the following. Everyone has to answer, and everyone has to answer right only, because it's not difficult question. Slowly. I already told you not to hurry. Okay, others, quickly. Okay, if you look into the given question very correctly, they are saying that First option, used in anabolic reactions. What we used in the anabolic reactions? Of course, we use the ATP in the anabolic reactions. And I already clearly told you what will be the energy storage molecule. The energy storage molecule is the ATP only. And see, this is the option most of the students will get confused. Coupled with the energy requiring reactions. What do you mean by that, friends? What do you mean by the word coupled with energy requiring reaction? What do you mean by this line? Which means which reactions that require energy, in that reactions ATP will be used. That is called coupled with the energy requiring reaction. There is a requirement of ATP. So obviously this is also right. Then when I break down the ATP, then there is a formation of ADP and PI. Correct, na? So A, B, C will be the 1 and 2 will be the D. Very good. Perfect. Perfect. Okay. Now, I already told you, friends, you people no need to remember any structure at all. Okay. I will tell you what to remember. Okay. Now you understand what is ATP also. Now we should understand what is NADH, what is FADH2, and what is coenzyme A. Because friends, these things we always use in our chemistry till the end of the lesson. So coenzyme A, you always know it. We called acetyl CoA. Correct na? You call melanoyl CoA. So what is this particular CoA? Coenzyme A. Remember everyone, the coenzyme A is made up of 3 individual structures. Don't worry about the structure. One is called phosphorylated ADP, pantothenic acid, amino ethane thiol. These 3 together make up of the coenzyme A. Now you have to understand, sir, why did you call phosphorylated ADP? What is the reason? There is a presence of 2 phosphates. That's why we call, and there is a adenine. So we should call ADP only. Then why do we call phosphorylated ADP? Because friends, the sugar at a third carbon, it is added by the phosphate group. That's the reason only we call phosphorylated ADP. No need to get confused with it. Now listen, whatever I am saying to all of you, remember as it is. No need to remember any equations, nothing. What every student has to remember, what is NAD plus, what is FAD, and what is FADH2, and what is their structures. This is also extremely important, friends. Remember my words. NAD plus, it is called, remember all the students, NAD plus is called as oxidized form. Okay, most of the people are having a confusion. NADH means, which is called reduced form. Okay, NAD plus is the oxidized form, NADH is the reduced form. All the students listen my words. Whenever you heard the NADH and FADH2, every student remember, in biochemistry, we called them as electron carriers. Please do not forget this particular statement till the end of the lesson. What NADH and FADH2 can act like? Electron transport chain. You all studied in the intermediate and degree and MSc level. We use it to say that NADH and FADH2 are transporting the electron to the electron transport chain in the inner membrane of the mitochondria. You heard that. We're going to read it. So this NADH and FADH2 in the reduced form only. In the reduced form only, they can act like a electron carriers. So friends, what exactly you have to remember? Their structure. You only look into this. NAD plus made up of ADP. You can see, right? It made up of ribose. And how many phosphates are there? 2 phosphates are there. Friends, and what does it contain? Everyone remember, it contains nicotinamide. Here N represents nicotinamide adenine dinucleotide. Friends, that is not the question. What is the question you can expect in the exam? The nicotinamide ring is derived from which amino acid? Sorry, which vitamin? This is the question we can get in the exam. What is that vitamin? Niacin. Nicotinamide giving rise to the niacin. And what you have to remember for the exam point of view, the most important point every student has to remember for the exam, NAD plus is involved. in the carbon oxygen double bond reaction. This is important for the exam. What I am saying means wherever there is a formation of the carbon oxygen double bond in the biochemical reactions, there NAD plus is involved. And the vitamin is niacin. And what about the FAD? So friends, remember everyone, the FAD, flavin adenine dinucleotide. Once again, what you have to remember, FAD is the oxidized form. And what will be the reduced form? FADH2 will be the reduced form. What did I told you just now? In the reduced form, it is acting as the electron carrier. Please don't get confused with that particular point. And friends, here what did I told you? NAD plus involved in the carbon oxygen double bond reaction. And friends, same, what you have to understand, FAD is involved in what? Carbon-carbon double bond reaction. That's an important point. That is in the carbon-oxygen double bond reaction. This is in the carbon-carbon double bond reaction. You will see in the upcoming slides in the enzymes. Now, what does the FAD made up of? You only look into this. Flavin adenine dinucleotide. Adenine, sugar, two phosphate groups, and the ring, which we generally call it as what, friends? Riboflavin ring, which is derived from the vitamin B2. You people has to remember compulsory, very good, which we can call the ribitol. So FAD will be the oxidized form, FADH2 will be the reduced form. Okay, and what is the function of acetyl CoA? CoA is the one which activate the acetyl group. See here, this is the CoA. To the CoA, there is an addition of the acetyl group. It is converted into acetyl CoA. Okay, don't be hurry. I want every one of you think it and has to answer the question. Okay, take your time. Take 1 minute of time, but make sure that everyone will give me the correct answer. In the question, oxidized form is also there. In the question, reduced form also there. Use your brain. Slowly answer the question. Everyone, only 3-4 people are answering. So fast. Don't type in that way. Give the entire answer at a time. Sunil. Slowly. Yeah, others. Stop thinking it's a difficult question. It's not difficult, right? Others. Just now I have explained it to all of you, right? Why are you taking this much of time? Look into this. What did I told you? NAD plus will be the oxidized form, which is involved in which reactions? Carbon oxygen double bond. Correct or not? What is FAD and NADH2, FADH2? You only try to understand. Now, what is FAD? Everyone tell me. What is FAD? I told you, right? FAD is the one which contain riboflavin. So will be the D. Correct now? This will be the A. What is NADH2? I told you NADH2 it is. No, sorry. No need to get confused. This is NAD plus and this is NADH2 and this is FADH2 and this is the FAD. First write it down. This is the oxidized form. This is also oxidized form. This will be the reduced form. This will be the reduced form and this will be the coenzyme A. You can easily answer coenzyme A, which is involved in transferring the acetyl group. So this will be the C. Correct now? And what will be the NAD plus? I told you it is the coenzyme A involved in the carbon oxygen double bond formation. Correct now? Then what will be this particular NADH plus? Don't get confused. This is the coenzyme after carbon oxygen double bond. I told you, right? Reducing equivalent, which is involved in the electron transport flow. Don't confuse with that. And what about the FADH2? You can understand FADH2 means reduced form of the flavin adenine dinucleotide. And what about the coenzyme A, which is involved in transferring the acetyl group? Easy to remember it. No need to get confused. Anyone given the correct answer? 1A, 2D. Very, very good. Very good. Very good. Very good. Slowly understand which is the oxidized and which is the reduced form. Okay. So why we studied this ATP, NADH and FADH2 is because all over the biochemistry we will read this. That's the reason. Okay. Now the real pathways are going to start from now. What exactly we have to study in the biochemistry is this particular slide. If you understand this particular slide correctly, 70% you will get a very good idea what exactly we have to study in the carbohydrate metabolism in our syllabus. You will get a crystal clear idea. Follow me, every one of you, very correctly. Now let's start it. Every student pay attention towards this. This is the one you have to understand very nicely. I already told you, friends, whether you take a uh, carbohydrate, ultimately it has to be converted into what? Monosaccharide. What is that monosaccharide? Especially glucose. Because friends, glucose only entered into the glycolysis, not any other not any other monosaccharide. That also you can ask me a very good question. Sir, why only glucose? That's also a very good question. Because friends, in the cytoplasm, there are 10 enzymes are present. Those enzymes can break down only glucose. It cannot break down fructose. It cannot break down galactose. What is happening to that galactose? What is happening to that fructose? They are actually converting into glucose. That's the simple point you have to remember. Okay, anyway, let me explain to all of you very briefly. Please pay attention towards this diagram very nicely. You have to understand. Look into the point. So first, glucose entered into the bloodstream. Now, what is happening? In every single cell, some students will think that glycolysis never takes place in all the cells, which is a wrong statement. So they can ask one statement in the exam: glycolysis is a limited process which is happening in few cells. That is wrong statement, friends. It is not like that. Glycolysis will occur in every single cell in our body, because every cell require energy, right? So glucose undergoes how many steps? It undergoes 10 steps, which we can call glycolysis, and going to give rise to the pyruvate. We will discuss it elaboratively. Glycolysis, right? Lysis is breakdown. Glyco is glucose. So breakdown of the glucose into pyruvate. That is what exactly we can call glycolysis. That is fine. Okay, listen, I will explain depth here. Now, what happens to this particular pyruvate, friends? Pyruvate, in the presence of oxygen, it is converted into acetyl-CoA, and acetyl-CoA enter into the citric acid cycle. And we all know very well, citric acid cycle give rise to ATP, NADH2 and FADH. We know that very well. And the same pyruvate, in the absence of oxygen, it is converted into lactate. I'm talking about only human being. Yeast we will understand later. So glucose converted into pyruvate. Pyruvate in the presence of oxygen give rise to acetyl-CoA. Absence of oxygen, it is going to give rise to the lactate. This every one of you know it. Now you tell me, just imagine, you have to think like that. Suppose I had a biryani, I had a huge meal. When I had a huge meal, I will get a lot of glucose, right? I will get an abundant amount of glucose. Suppose example, I had a chicken. So the chicken contain protein also, chicken contain a lot of glucose, carbohydrates, the rice also. Now you have to understand the point. Do you think how much glucose our body uses? Tell me how much amount of the glucose is required for the cell. It will only use that much glucose, right? It is not using the all the glucose that we eat. That is not possible. So it uses how much is required. Then what it is going to do, the excess glucose it is converting into glycogen. What? It is storing in the form of a glycogen. So what I am doing, tell me, I am synthesizing the glycogen. Every one of you look into the definition. Genesis, synthesis. What I am synthesizing? Glycogen I am synthesizing. So synthesis of a glycogen from the excess glucose. Every student will say that glucose. You should not use the word glucose here. You should always use the word excess glucose. From excess glucose only we are converting into glycogen. That glycogen only we can call synthesis of glycogen called as glycogenesis, where most of the glycogen can be stored in the liver, followed by the muscle, and little bit amount of the glucose can also be stored in the wonderful brain. Very little. Okay, so today I am having a lot of glucose, I converted into glycogen. So today I had a biryani, I have a lot of glucose in my body. Tomorrow what I am doing? Tomorrow I am doing fasting. I did not have any food from morning. I did not have any food. But you know very well, even though you did not have any food, you can survive easily. What is happening over there? The glycogen is getting breakdown and it is converted back into glucose. So what is happening now? Glycogenolysis. What is lysis? Breakdown. What is breaking down? Glycogen is breaking down. That's the reason only we can call glycogenolysis. Breakdown of the glycogen. And where the glycogenolysis will break down where the glycogenolysis occur in the liver, because liver is the king of metabolism. Liver is the area where the glycogen is stored. So breakdown also occur in the liver only. So glycogenesis, storing occur in the liver, breakdown also occur in the liver. Never forget that point. Okay, today I am doing fasting. So whatever the glucose that is present in my body, sorry, whatever the glycogen present in my body, that got breakdown. I have used it nicely, no problem. But friends, tomorrow also I am doing fasting. Every student remember, when the fasting crosses 24 hours, we don't use the word fasting anymore. We use the word harvesting. So after 24 hours it become harvesting. So now you tell me, how can I survive? You know very well, even though I don't eat 24 hours continuously also I will survive. What is happening? And you can ask me, sir, whatever the glycogen that we already stored, that also we used it, then how can we survive? Friends, now our body start producing the glucose from where? From the fat, from the amino acid, from the protein, from the lactic acid. Now you tell me, does this fat, amino acid, protein and lactic acid, are they carbohydrates? No, not at all. What do we call them? We call them as non-carbohydrate sources. What do we call them? Non-carbohydrate sources. That's the reason I am calling the name of the process as gluconeogenesis. Most of the people will don't understand why we have given the name gluconeogenesis. Tell me. Genesis, production. We are synthesizing something. What we are synthesizing? We are synthesizing glucose. Then why did we call neo? My friends, neo means new. What do you mean by the word new here? Other than carbohydrate. I hope you understand correctly. So I am synthesizing the glucose from lipids, fats, proteins, amino acid. Are these carbohydrates? No. So I am synthesizing the glucose other than the carbohydrates from the new sources. That is the reason only we can call gluconeogenesis. So friends, what are the steps you all people has to read here? We have to read glycolysis, breaking down of the glucose into pyruvate. We have to read glycogenesis, synthesis of glycogen from the excess glucose. Glycogenolysis, breakdown of the glycogen to produce glucose again. Gluconeogenesis, production of the glucose other than the carbohydrate source, that is called gluconeogenesis. All these processes we have to read very depth. Now in our exams, Most of the questions will come from where, whether it is the JL or DL or set exam. Every one of you remember, locations are very important, friends. Means in which cells, exactly at which location of the cell various biochemical reactions are taking place. So remembering them is extremely important for exam. So you all people started reading from the intermediate level. Glycolysis means it always takes place in the cytosol, not the cytoplasm. You should always remember cytosol. Cytosol means there is no organelles. It is only a fluid. Why only in the cytosol? What is the reason? Because glycolysis requires 10 enzymes. All the 10 enzymes are exclusively present in the cytosol only. That's the reason it is happening in the cytosol. Now, friends, glucose giving rise to pyruvate. Every student will get a super confusion that I was repeatedly saying to all of you. Friends, pyruvate is converting into acetyl-CoA. 90% of the students will forget where the pyruvate is converting into acetyl-CoA. That's a very good question. You know, friends, we will study upcoming slides. Pyruvate converted into acetyl-CoA. That process called as pyruvate decarboxylation. What is that? Pyruvate decarboxylation. Where it is happening? That's also a very good question. Pyruvate converted into acetyl-CoA in the mitochondrial matrix. Why? That's also a question. If the pyruvate wants to convert into acetyl-CoA, there is a requirement of enzyme called as dehydrogenase enzyme complex. That enzyme complex also present in the matrix only. That's why first pyruvate enter into the mitochondria. In the mitochondrial matrix, it is converted into acetyl-CoA. Then acetyl-CoA enter into the citric acid cycle. What students will think? First pyruvate enter into the citric acid cycle. Then in the citric acid cycle, pyruvate converted into acetyl-CoA. That is wrong statement. Pyruvate cannot enter into the citric acid cycle. First it entered into the matrix, converted into acetyl-CoA. Acetyl-CoA entered into the citric acid cycle. So the site of location of conversion of pyruvate to acetyl-CoA is also matrix only, and where the Krebs cycle or the citric acid cycle is taking place, that is also taking place in the mitochondrial matrix only. Why? All the enzymes required for the citric acid cycle present in the matrix. And after the citric acid cycle, we are having electron transport chain. Where the electron transport chain is taking place? Electron transport chain is taking place on the inner membrane of mitochondria. So you all know very well, I told you in the cell biology also, this is the outer membrane and this is the inner membrane. Inner membrane can be thrown into foldings. You all people know it very well. You can call cristae. Now you have to ask me a question, why? Why the electron transport chain has to happen in the inner mitochondrial membrane? Why not in any other place? All the students remember, inner mitochondrial membrane contain 4 electron acceptor complexes. How many? We will study 4 electron acceptor complexes. And moreover, the inner membrane of mitochondria having a lot of surface area. Surface area is maximum. So all the electron acceptors are accumulated or embedded in the inner membrane of mitochondria. That's why electron transport chain is taking place over there. Then friends, next step we can call chemiosmosis. You can also call, friends, oxidative phosphorylation. Oxidative phosphorylation is the process where we are going to synthesize abundant amount of ATP. And where it exactly occur? It occur between inner membrane to intermembrane space. You know, right? Between the outer, between the inner, what space it is? Intermembrane space. So exactly between the inner membrane to intermembrane, this process will occur. It is mandatory, friends. You people has to remember compulsory where all these reactions are taking place, right? So in the tomorrow class onwards, we will study the first step of the carbohydrate metabolism, which is the glycolysis. So remember, I want to finish everything very fast. So I will go little faster from tomorrow, but I'll explain very effectively. Okay. So, and I'm telling you, first few days, everyone will show great interest in the competitive exams. But trust me, if you want to crack this exam, You have to show your consistency. Remember my words, in order to crack the competitive exam, you do not require to be an intelligent person, but you require to be a consistent person. See, many of you are very intelligent, many of you know the concept, but if you never practice, you're never successful. It doesn't matter how intelligent you are. So work sincerely for the 60 days without missing even a single day also. Obviously, we can crack it. Okay. Is it clear, everyone, so far till this particular slide? We will continue tomorrow on the same time on the biochemistry. Everyone say yes or no. Is it clear? Quickly, quickly. Okay, okay. I will be sending the molecular biology class link right now. I want every one of you, please join sharp 6:30 a.m. right now. I mean to say after half an hour, the molecular biology class. Okay. We will go a little faster. Please join sharp 6:30 a.m. Okay. Make sure that your audio and video will be turned off. Okay. I'll see you at 6:30 a.m. sharp. Okay. Thank you. Thank you all. Don't sleep in the middle. Thank you.